Good afternoon, and thank you for joining this Alcyomics webinar in association with the Drug Safety Testing Centre, the DSTC Japan. I'm Asif Tula, and I'm the Business Development Manager for Alcyomics. Today, I'm going to talk you through how Alcyomics can tailor their human-based scientific services to provide novel testing solutions for the development of therapeutics towards SARS CoV-2. I also have Professor Anne Dickinson, the founder and Chief Executive Officer of Alcyomics, with me to answer questions at the end of this presentation. If you have any questions, please type them in the message bar on the webinar platform. Alternatively, if you have any questions after the webinar, you can send them to us via the email addresses on the screen. I will commence by providing an introduction to Alcyomics and the Skimmune technology. This was covered in detail in a webinar presented in April and is available on demand on the Alcyomics YouTube channel. I will then move on to describe the suite of human-based assays that we have available for COVID-19 testing. These are to test the immune responses to vaccines, novel or repurposed drug candidates, and to aid the discovery of new drug targets and for safety and efficacy assessment of drugs. I will now go on to give an introduction to Alcyomics and our Skimmune technology. Alcyomics is a biotechnology company based in the northeast of England in Newcastle upon Tyne. We have been trading since 2009 and offer a unique service provision based on preclinical product safety testing. Our tests are based on human in vitro skin tests, which we call Skimmune. And Skimmune has been developed from more than 20 years' experience using the skin explant model in the research setting. Skimmune is a non-animal alternative model and supports the 3 Rs, the reduction, refinement, and replacement of animals in research. Alcyomics developed Skimmune to help our clients identify adverse drug reactions before they take their products into the clinic. We can help you reduce risk and cost in the drug development program by the early identification of potential adverse events. Skimmune is a unique human in vitro model, which we have shown through our published data to be highly predictive of clinical classification of adverse reactions and is applicable to all drug types, such as small molecule drugs, biologics, including monoclonal antibodies, their aggregated formats, biosimilars and biobetters, nanomedicines, DNA-based therapeutics, and also cellular therapies. The Skimmune platform predicts hypersensitivity responses to pharmaceuticals, including small molecule drugs and biologics, such as monoclonal antibodies, as well as cosmetics and chemical compounds. It is a human in vitro test which uses fresh human skin and blood from the same individual in an autologous setting. This next slide describes the Skimmune assay for the assessment of small molecule drugs and chemicals and compounds. We can obtain skin and blood from a healthy volunteer. We can extract the immune cells from the blood to make monocyte-derived dendritic cells. We then allow the test compound or the test drug to present itself to the immune system by interaction with these monocyte-derived dendritic cells. The monocyte-derived dendritic cells are then co-cultured with autologous T cells. If the test article activates the immune system, the T cells will be primed such that when co-cultured with autologous skin from the same individual, the T cells will release cytokines that cause damage in the skin at the dermal epidermal junction. This damage can be graded using a clinical grading system called the Lerner classification, which has been used in clinical practice. These SCIMMUNE assays are ideally suited to the assessment of adverse immune reactions to small molecule drugs, protein molecules, and the assays are used to determine hypersensitivity reactions and ensure drug safety. It's important to note here that although skin tissue is used in the assay, this is a predictive marker of the systemic immune response. Therefore, the damage observed in the skin tissue is indicative of a systemic immune reaction and is not specific or localized to the skin only. We grade the slides using a clinical classification called the Lerner classification. This has been used in clinical practice for several decades and represents a clinically validated system that we use to grade the skin sections. In Alcyomics, all slides are graded by 2 independent histopathologists in a graded fashion. Grade 1 on the left shows normal healthy skin tissue, and this represents a negative response. The compound is not causing a reaction. The next image along shows a grade 2 positive response. Here we can start to see small vacuoles forming along the dermal epidermal junction. This would appear as a skin rash in the clinic. The next response is a grade 3 positive response. This is slightly more severe, and we start to see the small vacuoles forming larger clefts along the dermal epidermal junction. We also start to see necrosis forming in the upper epidermal regions. This would present as skin blistering in the clinic. The most severe response on the right is grade 4 positive response. Here we see the complete separation of the epidermis from the dermis due to the breakdown of the connective proteins, and it appears as though the skin is falling off. For the assessment of novel drugs, chemicals and compounds, Alcyomics also provides screening assays for the early identification of adverse events. On this slide, we show T cell proliferation assay on the left and interferon gamma on the right. These assays are useful for screening compounds quickly, and this is particularly important during the development of novel therapeutics for COVID-19. Although cell-based assays such as this can show variation between donors, they are useful for a quick screen to aid drug discovery, and the lead compounds can be assessed with greater accuracy in the SCIMMUNE assay further on in product development. While our assays typically require less donors to identify compounds as being a problem or not when compared to other assays on the market, the use of SCIMMUNE, due to the addition of the skin tissue and the histopathological readout, makes it more easy to identify compounds as sensitizer or non-sensitizer due to the specificity of the response causing damage to the skin tissue. We have developed these assays to bridge the gap between preclinical research and animal studies and first-in-man clinical trials. SCIMMUNE represents a human in vitro test that has been shown to be predictive of the drug response with a high degree of predictability to their clinical classification. Having now provided you with a quick recap of Alcyomics' current service provision, including our novel SCIMMUNE platform, I will now progress to describe our capability specifically towards COVID-19 drug development. In response to the global effort towards COVID-19, we tasked our scientists with repurposing our human-based assays to develop a suite of tests which could be used to aid efforts to identify new drug targets towards SARS CoV-2 and expedite the safety and efficacy assessment of these new vaccines, drugs, and also repurposed drug candidates, which I will now describe to you. A bit of background for COVID-19. The incubation period for the virus, SARS CoV-2, is 3 to 7 days. 80% of infections are mild or asymptomatic, 15% are severe, requiring oxygen, and 5 are critical. The virus uses the host cell receptor ACE2 to infect the human airway epithelial. The spike and nucleocaspid proteins have been shown as the most immunogenic expressed proteins during infection. Specific T-cell responses against the SARS-CoV-2 virus is important for recognition and killing of infected cells, particularly in the lungs. The cytokine storm produced in susceptible individuals is a severe systemic inflammatory response, releasing pro-inflammatory cytokines including interferon gamma, IL-1 beta and IL-6, and chemokines including CCL1, CCL2, CCL5, CXL8, CXL9, CXL10 and others. There are differences in levels when comparing severe versus mild infected patients. I will now describe the suite of assays that we have developed at Alcyomics. These were developed to answer the following questions. Do you need to test assays for immune responses to vaccines or novel therapeutics? Are you looking for new drug targets? Do you require safety and efficacy testing for COVID-19 vaccines under development? Do you require immunomodulatory inhibitors for SARS-CoV-2? Do you want to test new or repositioned drugs to see their effects on SARS-CoV-2 immune response? If any of these points apply to you, then you can partner with Alcyomics to aid drug discovery and development pipelines. We use human-based assays, therefore no animal tests, to ensure there are no interspecies differences when analysing our assays. This can help you to progress product development with quality, reliable data, ensuring accurate go, no-go decisions. The rationale for the service provision that we provide at Alcyomics. Understanding the immune response to the SARS-CoV-2 virus is critical for the evaluation of vaccines and quality management of patients. The cytokine storm that occurs in the most susceptible individuals is a serious systemic inflammatory response, releasing several pro-inflammatory cytokines and chemokines. A better understanding of this in patients is necessary for the development of novel therapeutics, clinical management, and successful clinical trials. The next section will focus on evaluating the immune response to vaccines and novel therapeutics. This assay, like the screen assay, uses fresh human blood from an individual and can be used to assess how the vaccine or novel drug is activating the immune system. We produce monocyte-derived dendritic cells, which are then incubated with the test vaccine before being washed. This allows the test article or the test vaccine to activate the immune system. The washed mature dendritic cells are then co-cultured with T cells from the same blood donor. If the dendritic cells are activated, this will prime the T cells, which will proliferate and release cytokines and chemokines, which can be measured. At Alcyomics, we use multiplex cytokine and chemokine arrays, as well as tritiated thymidine incorporation assays to measure T cell proliferation responses. The readouts that we generate here include cytokine and chemokine profiles, T cell proliferation responses, TC and TH subset profiles, and mRNA expression analysis on activated dendritic cells. This next slide will focus on identification of new drug targets. Using our in-house screening assays, Alcyomics are able to detect immune responses to SARS CoV-2 antigenic spike and nucleocapsid proteins and assess cytokine and chemokine release. TC and TH ratios to determine immune response using a range of HLA peptides. RNA-seq and mRNA expression analysis can be used to help identify novel drug targets. The key objective for SARS CoV-2 novel drug discovery and testing is to use our proprietary technology to test virus peptides identified from spike and nucleocapsid immunogenic proteins associated with HLA class 1 and class 2 epitopes to discover novel drug targets. We can assess the immune response to peptides via immune activation in vitro using HLA-typed human PBMCs. From this, we can generate cytokine, chemokine, and RNA expression profiles, which can then be correlated with epidemiological factors, HLA genotypes, and also common genotypes from SARS or MERS disease. This will provide disease signatures for this drug discovery and responder profiles. This information will help inform the cytokine storm response, identify pathways of immune activation, and identify differences in susceptibility to COVID-19. Validation studies can then be completed with patient samples from COVID-19. All this information is valuable for drug discovery efforts to design therapeutic strategies to modulate the immune response and vaccine development. The activation of dendritic cells with SARS-CoV-2 spike and nucleocapsid proteins can allow the identification of DC activation pathways and novel drug targets via RNA-seq and RT-qPCR. These results can then be correlated with T-cell activation and cytokine release. Novel drug targets or vaccines can be tested to determine the immune response and to show inhibition of major pathways using alterations in RNA-seq profiles. This next section will focus on the safety and efficacy testing for COVID-19 vaccines under development. These assays are the normal routine assays that Alcyomics provide to companies in the pharmaceutical industry globally. Our approach is to show the specificity of the vaccine against antigens rather than antigens cross-reactive on bystander cells and exacerbated release of cytokines. Our novel expertise uses Alcyomics T-cell specificity assays as well as our patented SCIMMUNE assays. These are used to assess safety and efficacy. Typical readouts include antiviral specific T-cell readouts, as well as histopathological analysis from the SCIMMUNE assay. We have previously published our work on antiviral T-cell analysis, and this is available with this reference on the bottom of the screen. If you would like a PDF copy of this, please feel free to ask via email. This next section is looking at immunomodulatory inhibitors to SARS-CoV-2. We can use our immunomodulatory or immune inhibition SCIMMUNE assay to look at the efficacy of immunomodulatory peptides and proteins. In this assay, we start with a grade 3 positive response by the induction of damage through an MLR, a mixed lymphocyte reaction. We can then add the test monoclonal antibody or protein at different concentrations to try to reduce the damage from grade 3 down to grade 1. In this example, you can see that adding the test protein at 0.15 microgram per mil reduces to grade 2, and increasing the concentration to 1.5 microgram per mil brings us down to a grade 1 negative response. So in summary, I just wanted to explain how working with Alcyomics can aid your safety testing of novel therapeutics towards COVID-19 and also drug development efforts with respect to looking at novel drug targets and also repurposed drug candidates. If you need assays to test immune responses to vaccines and novel therapeutics, we can use our standard patented Alcyomics immune platform for the testing of immune responses to vaccines and novel therapeutics. If you are looking at identifying new drug targets, we can also use the SCIMMune platform with SARS CoV-2 spike and nucleic acid proteins to induce immune responses and investigate the pathways and networks involved to identify drug targets. This uses RNA-seq, mRNA expression analysis, multiplex cytokine and chemokine profiles, and this can help to investigate the mode of action. If you require safety and efficacy testing for COVID-19 vaccines under development, we can use antiviral T-cell specific assays alongside our SCIMMune platform and demonstrate safety and efficacy of these antiviral vaccines. If you require testing of immunomodulatory inhibitors towards COVID-19, or you want to test new or repositioned drugs to see their effect on SARS CoV-2 virus immune responses, we can assess the efficacy of drugs to suppress the cytokine storm and use our immunomodulatory or immune inhibition SCIMMune model, as well as inhibiting T-cell responses in the assessment of drugs. This brings us to the end of this short webinar, focusing on testing solutions for the development of novel therapeutics towards SARS CoV-2, including vaccines, drugs, and repurposed drug candidates. I would like to thank you for your time and for attending this special webinar in association with the DSTC, the Drug Safety Testing Center in Japan. We have a few minutes